Okay, hi again everyone and welcome again to Biology 2. So this time around, this is the last video for the module number 1, which is Mendelian inheritance, and we'll be talking about dihybrid crosses right now. So in dihybrid crosses, the thing that you have to take note is that you are no longer taking note of only one gene pair or one allelic pair. You are going to be taking note and do crosses using two or more gene pairs. So that's the basic idea here. So for a single trait, it's called monohybrid crosses. But if you have two traits, it's called dihybrid crosses, so on and so forth. So if you have three gene pairs, you have trihybrid crosses, and so on and so forth. As you can see in the example here, the cat has two gene pairs here, the S and the B, crossed with an S and also the B. So we'll be talking about that a little bit later. Okay, so how do you do crosses with dihybrid crosses? So this will be the steps that you need to take note. Of course, first of all, you determine the parents' genotypes. So that's why we studied about the law of segregation. So you already know how to get the genotypes. And then after that, determine the possible gametes that can form when alleles segregate. So in this case, in monohybrid crosses, you just have to separate them. But in this time, you will be taking note of the law of independent assortment, wherein the two gene pairs, you will be combining them so that you will be forming the unique gametes of each parent, wherein there will be two letters present. The first letter of the gene pair, first gene pair, and the second and the other letter of the second gene pair. So place number 3 or the third step will be to place each gamete combination two different letters like big A and big B, small a and big B, or small a and small b for one parent over each column of the cross. So it's going to use the Punnett square at first so that we will be able to really see the principles behind that. And then number 4, do the same for other parents to the left of each row of the 4 by 4 crosses, 4 by 4 cross or the Punnett square. So you will be noticing here that for monohybrid cross, you have only 2 by 2. But for a dihybrid cross, you will now have a 4 by 4 Punnett square, which will be yielding 16 progenies in the Punnett square. Let's look at that. Okay, so for example, you have this dihybrid cross. You have purple flower and white flower and tall and dominant. So these are actually the same traits that Mendel made use of in his experiment. So for this one, the purple, capital P, is the dominant one, while the white, which is recessive, is the smaller letter or the small p. On the stem length, we now have here the tall, which is the capital T, which is the dominant, and the small t, which is the dwarf, which is the recessive. So how do we make use of this information for your dihybrid crosses? So let's start first with the parentals. For example, you have a homozygous tall and purple plant or homozygous parent for both of these gene pairs, and we have also another parent which is homozygous recessive for both the stem length and also for the colors, flower color. So you have here tall and purple, and the other one will be dwarf and white. So the question is, how are you able to get the F1 generation? Okay, so this is how you will be getting that. First of all, get the gametes of the parents. Let's start first with the first parent, which is homozygous dominant for T, homozygous dominant for P. So big T, big P, big P, big P. First of all, get the first gene pair and separate them to get the unique gametes. Get the second gene pair and get the unique gametes that you have. So you have here a big T and a big T and a big P and a big P for the gametes. Then you're going to be combining them together to find the unique gametes produced by this particular parent. So how do you do that? You get the big T over here, the big T over here, and randomly distribute the big T and the big P, as you can see here. Then you will have we will have here the gametes big T, big P, dominant for TNP, dominant for TNP, dominant for TNP. So these are the gametes that will be produced by the first parents. And of course, how many types of gametes? You only have one type of gamete, so it's quite easy to get that. Okay. Now the next thing you're going to do is to get the gametes of the parents. The same procedure. First gene pair, you just separate this into small t. And the second gene pair, you just separate this into small p and small p, and then you're going to randomly combine them together because of the law of independent assortment. So they independently assort. So you have the small t and the small p here, and then you're just going to combine the small p and the small p, randomly distribute to produce the gametes that will be formed by the second parent. Now, of course, it only has one kind and there's only one type of gamete. So now, how do you actually do the process here? So we have here the long stem and the purple, the short stem and the purple. So we have the this genotype as mentioned already. So let's get to it. Let's how do we get that? Combine the gametes of the first parent with the gametes of the second parent. So in this case, if you're going to make use of the Punnett square, all four of them. So all four of this will be on one side, all four of this will be on the other side, and of course combining them together, this will be a little bit tedious, but of course you will be needing this in order to get the children or the progeny. So all of them you will be noticing would be big T, small t, big P, small p, or heterozygote. So that will be the answer to this one. 100% will be heterozygote at this point in time. Now we can make it a little bit shorter since we already know that there are only two T's. So you just write it 2T here, and then you have two P's, you write it 2P over here, and then you can get actually the progeny, which is the 4TP, as I mentioned a while. So this is a shorter version of what we've done a while ago. On the other parent, you will also do the same thing to make it shorter. Since there are two of these small t's, then you write 2t, small of these 2p, you combine them together, and then you will produce 4 small t, small p as gametes. And to make it a shortcut, to make it faster, a while ago we made the Punnett square 16 panes. It will be quite simple if you just do like this. So you just have 4 of those big T's, big P. You have 4 of those small t and small p. You just simply combine them together in one pane, and then what will happen here? So you will have big T, small t, big P, small p, and you just multiply the numbers together. 4 times 4, you get 16, and then you get the same answer with a shorter duration of time, just making use of the shortcut. Okay, so you're now able to get the F1 generation. Now, what if we want to get the F2 generation? So in the F2 generation, you're going to cross together an F1 and another F1, and what would be the progenies produced by the F2 generation? Heterozygous. Okay, this is a little bit wrong, but we will get the answer to this one. Okay, let's get to it. So this is the hybrid cross that produces the F2 generation. First of all, you will allow the F1 and another F1 to self-pollinate. And this is how you get the gametes of the parent. So you have here the big T, small t, big P, small p, which would be the heterozygote, the F1 generation. First gene pair, you separate them again. So it's a big T and a small t. Second gene pair, you separate them. Supposedly big P ito. So you will get a big P and a small p over here. And then you just randomly distribute the big T and the small t. And then you also randomly distribute the big P and the small p. Just like that. And you will get here the gametes. Big T, big P, big T, small p, small t, big P, small t, small p. And then how many types of gametes? You will now here have 4 types of gametes. Now, to get the dihybrid cross, again, what you'd simply do is to combine this. So we already know that since both of them are heterozygotes, so one of the first the one of the parents you'll be putting on the top of the Punnett square, while the other one you're going to be putting on the right side, and then you're going to combine them together. So looking here, it becomes big T and big T, big P and big P, big T and big T, big P and small p, so on and so forth. So you're just going to combine all of these panes together, and it will be it's a little bit quite tedious, but if you want to do it like this, you can do it like that. Okay, so you already have this one. So there are 4 phenotypes that will be observed: a tall white, a short purple, and short white. So notice this one, the first pane, tall and then purple, tall and then purple, tall and then purple, tall and then purple. The next one, tall, purple, tall, and then now you have the white. It's a tall white, tall purple, tall and then white, tall and then purple, tall and then purple. In this case, you have a small t. It now becomes short and purple, and then short and purple. This will be tall and purple, tall and white, short and purple, and short and white. So you will have 9 tall and purple, 3 tall and white, 3 short and purple, and 1 short and wide. So that's how you're going to be doing that. So the ratio for a heterozygote by a heterozygote when there is a dihybrid cross would be a 9 is to 3 is to 3 is to 1 ratio. And actually, this is one of the automatic ratios that you need to remember. A hetero for both gene pairs that will be crossed with a second pair that is hetero for both gene pairs as well will always be yielding 9 is to 3 is to 3 is to 1 phenotypic ratio. Okay, you can actually make it a little bit faster when you make use of the fork method. So you just distribute first and first, first and second, second and first, and second and second. So you get here the answer here. Big T, big T, big T, small t, big T, small t, and small t, small t. On the other hand, the other gene pair, you just combine it like that. Or before that, the genotypic ratio of this first gene pair is 1 is to 2 is to 1. And then you combine this, the first and first, big P, second and second, supposedly here, big P, small p, and then you have here the second and the first combined, supposedly here, big P, small p, and finally the last and the last, it will be small p, small p. So the genotypic ratio is also the same, 1 big P, 2 big P, small p, and 1 small p. Now, you can make use of this genotypic ratio in order to get the genotypic ratio of the whole dihybrid cross. So that's the first gene pair and the second gene pair. So how do you do that? First of all, write down the first gene pair. I'm gonna put it here. Okay, it's a little bit complicated. Okay, let me just finish this up. So that's how you're going to do that. Yeah. First of all, you're going to distribute the genotypic ratio of the first gene pair that we got a while ago. So you have 1 is to 2 is to 1. So since there are 4 children, you have 1/4 of the big T, 2/4 of the big T, small t, 1/4 or the small t, and that will be it. Then you're going to distribute the genotypic ratio of the second gene pair, which is the same as well, 1 is to 2 is to 1. So you have your 1/4 big P, 2/4 big P, small p, 1/4 small p, and you distribute that to all of the possible combinations of the first gene pair. So you combine this, and then the big T, small t, combine all of the 3 of the second gene pair, and the small t combine all of the the 3 of this in the second gene pair. And simply combining them together, you will get all of the possible gametes and their frequencies as well. So in this case, big T, go here, big P, and you have the big T, big P, and then you're just gonna multiply. The numerator times the numerator is 1, denominator times the denominator, 4 times 4 is 16, so you get 1/16, big P. Okay, and another one is you combine this one and the second, so it becomes big P, small p, and the frequency will be, the proportion will become 1 times 2, it's equal to 4 times 4 is 16, 2/16, so on and so forth, just to make it simplified. So you will be noticing here that this will be all the genotypes produced by that one. And notice you can just simplify as 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1 ratio. Okay, so a hetero by a hetero, in terms of genotypic ratio, you will always be producing 1 to 1 to 4 to 1 to 1 genotypic ratio. Hetero for both gene pairs, cross with a parent who is hetero for both gene pairs as well. Okay, so now you have here the genotypic ratio, as already mentioned a while ago, and you already also know what we call as the phenotypes, 9 is to 3 is to 3 is to 1 ratio. You can actually get the phenotypic ratio in a shorter way by making use of the fork method. So again, just get the genotypes for the first gene pair, and it's the phenotypic ratio will now become tall, and short, and then the second gene pair as well, which is the first. Combine with the second, combine this, and then combine, and you will get phenotypic ratio of 3 purple is to white. So this will be purple, and then white. So you'll notice this is 3/4 tall, 1/4 short, 3/4 purple, and 1/4 white. Making use of the phenotypic ratio, it will not be easy. And how do you do that? Okay, let me just give it all over again. And okay, to make it simple. So you already know that there is 3 is to 1 ratio for the tall and the short, 3/4 is to 1/4. So you just write it out like this. And then after that, you will be distributing the phenotypic ratio of the flower color. So we already know 3:1 ratio. So you have a 3/4 for the purple and 1/4 for the white combined with the 3/4 tall, and the 3/4 purple and the 1/4 white combined with the 1/4 short. Combining them all together, you will get the answer. 3/4 tall and purple, so you get tall and purple. 3 times 3, which is the numerator, is 9. 4 times 4, you will get a 16. So it's 9/16. On the other hand, tall going to the white, so you have a tall white here. And then 3 times 1 is 3 as a numerator. 4 times 4 is 16 for the denominator. 316 tall and white. The same thing here. Short and purple, 1 times 3 is 3. 4 times 4 is 16. So you get 3/16. And then 1 times 1 is 1. 4 and 4 times 4 is 16. So you have a 9:3:3:1 ratio, which is a little bit faster compared with the Punnett square. So that's how you're going to be doing that when you're being asked for the phenotypic ratio. Okay, so the last thing we need to talk about is what is a test cross and a back cross. Well, a test cross is simply, if you read this word, test cross is the cross between an unknown genotype and a homozygous recessive genotype. So you're going to cross the homozygous recessive with the unknown type, and this actually helps to determine the genotype of the unknown parent. The back cross, on the other hand, is a cross between the F1 hybrid with any of the parent, and it will be mentioned in the problem which parent are you talking about? Is the first parent or the second parent? Mostly, more likely describing the characteristics of the parents that needs to be back cross with. So F1 with any of the parents. Now, the importance of this is a back cross helps to recover useful characteristic in parental population. So just an example for the test cross. For example, you have here this kind of cross. You don't know the one parent, but you know it is dominant. While you're going to test cross this with the homozygous recessive. So depending on what will be produced, you can know the genotype of the unknown parent. If you get 100% purple, then you know that the genotype is big P and big P, or homozygous dominant, because it will be producing all heterozygotes if crossed with a homozygote recessive. So that's the only thing that can be produced with 100% purple flowers. But if you get a 50-50, 1:1 ratio for the white and the purple, then you know that the unknown parent is a heterozygote, because that can only produce two purples and two purples and two whites. So you now know that the unknown heterozygote is unknown parent is a heterozygote. And that ends our discussion this time around. So in the next video, we will be starting with the non-Mendelian inheritance.